Hello students, welcome to the crash course of biology. Till now we have discussed around 37 questions. Now it's time for the next question. So as I have promised that in today's session I'll be talking about the new chapter, which is human health and disease. As per your syllabus, as per your pattern is considered, see, CBSE board, that doesn't give us an idea that this chapter is important, this chapter is not important. On the basis of the last few years, the past few year questions, we generalize that these topics, they are important. But as per the CBSE, every chapter is important. So I have tried to incorporate, I have tried to include many questions which I have seen that is being repeated from the last few years. So only those questions we will be talking in the crash courses. It doesn't mean that you have to completely rely on these questions. I know these questions which I have included in the session, they'll give you a good marks, right? But for attaining a best marks, but for attaining highest marks in your examination, because your paper is for 70 marks, at least by reading all these questions, by throwing through all the syllabus, like I have included the most important topics, I'm 100% sure that you will be able to attain at least 50 marks. Right, and for attaining 50 to 60 marks and for getting a marks like 65 for the 70 marks, what you have to do, you have to thorough the whole syllabus, right? Now let's move on to the topic. In this chapter, in this session, I'll be discussing some questions from the chapter Human Health and Disease, and also we'll be discussing questions from microbes as well as plant breeding, right? Now, first of all, as you can see that this chapter is related to cancer. This question is related to cancer. So cancer is from the chapter Human Health and Disease. Human Health and Disease is a very, very, very important chapter. Various diseases are there which can be asked in a 5-mark question, such as AIDS is there, such as malaria is there, such as cancer is there. These 3 should be there in your pockets. Right? I don't mean to say that these diseases should be there in your pocket. I mean to say you should have a thorough knowledge, and that knowledge should be there in your pocket. This I mean to say. Along with that, what are bacterial disease, what are viral disease, what are fungal disease, these list of diseases that should be there in your pocket if you want to attain a 70 out of 70 marks in your examination. I'm 100% sure that you will be able to attain if you follow all my instructions. In the session, in the video sessions, not in this crash course, in the detailed session of CBSE 12th class, I have discussed regarding these different type of disease and their vector, right? We have discussed everything in detail. I have provided you the list of diseases. If you look at our ebooks, in ebooks also one table is mentioned. So you can follow the same from that ebooks as well. Like if you don't have any list of it, what you can do, you can download our ebooks. In our ebooks, there is the one column is present, the 2 columns are present. On one side, there are the names of the bacteria or the agents, and later on I have written the disease. So that is also very important, which can be asked in 1 mark. So that also you should keep in your pocket, right? So as you can see that this question is related to cancer. Like what all the things which are important as per this cancer is considered. First of all, what is cancer? Second thing is, what are the properties of the cancer cells? What are the diagnostic? Everything I have included over here. In this question, everything is included which is related to cancer. See, question I can put in a direct way as well, like what is a cancer and how these cells proliferate, what is the function, what are the properties of the cancerous cells, how we can prevent it, how we can detect it. A direct question can be asked, but that will be very much easy for you. You can easily attempt it. But over here, I have remolded this question so that you can have a clear idea that such type of questions can be asked so that you can get prepared for that. Now, as the question 38 is considered, let's move on to the question where I am reading the first part. Cancer is one of the most dreaded disease that we know. Explain the contact inhibition and the metastasis with respect to disease. Now, as per the metastasis, as per the contact inhibition is considered, these are the properties of the cancer cells. The first property is that they are rapidly dividing cells. They divide at a very fast rate. Second thing is they are immortal. They do not die. When we talk about a death of a cell, yes, cell do die, which is replaced by a new cell, right? This normal happens in our body. But as per the cancerous cells are considered, they are immortal. They do not undergo the process of apoptosis. Apoptosis is what? Programmed cell death, right? Contact inhibition is also one of the property of a normal cell. Let's take a culture of an animal cell. They form a unilayer of cells. Do not allow the overgrowth of cells. This is called as a contact inhibition when neighboring cell doesn't allow the cells to overgrow. This is called as a contact inhibition. On the contrary, to talk about the cancerous cells avoid this contact inhibition, right? They divide at a very fast rate. Second point which is written over here, as you can see, which is a metastasis. So what is a metastasis? That means they are being metastasizing. Metastasizing, that means cells, when we talk about a cancer, there are two types of tumors. One is called as a benign tumor which remain at one particular position, and second is a metastasis, in which they are migrated to different part of the body via blood and lymph. Just imagine a situation, the cells are migrating via blood, they are migrating via lymph to the various organs. They will divide over there as well, they will cause destruction over there as well. That is called as a tertiary stage, or the last stage of the cancer. That means now the cancer is metastasized. Now, as per the tumor is considered, we can easily eradicate the tumor because they are localized, they are present at a particular position. With the help of radiation, with the help of chemotherapy, we can remove it, the chances. But when it becomes metastasized, the chances of their removal is very, very, very less. Because the cells, they are very small, migrated to different positions, they can cause destruction at each and every organ. So that is why it is considered as a fourth stage. So both the property, contact inhibition, that means neighboring cells doesn't allow the cells to overgrow, right? Second is a metastasis, that means to metastasize or to migrate to various structure via blood and lymph. Right? Hope these are the two properties. Is it clear to you? Let's move on to the second part. Name the group of genes that have been identified in the normal cell that could lead to the cancer, how these genes causes cancer. Now, second part. So, second part of this question states that we have to discuss the genes. Yes, the genes of cancers are present. In a normal cell, oncogenes present as proto-oncogene. Present as proto-oncogenes, right? Now these proto-oncogenes, these are the double O. So proto-oncogenes which are present in a normal cell. So what are these? These are present in normal cell. Not only the proto-oncogene, along with that, the tumor suppressor genes are also present. Proto-oncogene and the tumor suppressor genes, they are also present. When they are activated by some physical agents, when they are activated by certain chemical agent, when they are activated by biological agents, it causes cancer. Now listen very carefully. Proto-oncogene, tumor suppressor gene normally present in a normal cell. Suppose some physical agents are there, suppose some chemical agents are there, suppose some biological agents they are acted on or they are being acted on such type of gene. In that condition, what happens is these proto-oncogenes and these tumor oncogenes, they get affected. Proto-oncogenes which are present in an inactive form, they will be converted into oncogene. That means what has happened over here? There is a gain of function. Gain of function. As per the tumor suppressor gene are considered, these are normally present and they suppress the tumor. If we talk about, there is a loss of function. Because of various agents like physical agents, chemical agents and the biological agents, tumor suppressor gene will be converted into tumor genes. And once these are present in our body, we will be affected with cancer. Right? Is it fine? Let's move on to the third part of this question. Name any two techniques that are useful in detecting cancer of internal organ. You all are acquainted with the CT scan, you all are acquainted with the MRI. These are the two techniques which is used for detection of cancer in the internal organ. So, CT scan. What is the full form of MRI? MRI is magnetic resonance imaging. Right? The CT scan means computed tomography. Magnetic resonance imaging and computed tomography, these are the two methods by which we can detect the cancer in internal organs. Why are cancer patients often given the alpha interferon as a part of treatment? Yes, nowadays various research are going in this direction. Because when we talk about the cancer cells, they are avoided by our immune system. They cannot detect it as that they are a normal cells, or they detect it as a normal cell only. Right? For that, no as such antibodies, they are present in our body. And alpha interferon treatment modify the immune system, modify the representation technique, due to which those cells or the cancer cell will be considered as a foreign cells, and they will be killed by our immune system only. Right? So, this was the fourth question of fourth part of the question number 38. Let's move on to the next question, that is a question number 39. So, read this question. By the time, let me prepare this board for you. Now, why do the symptoms of malaria not appear immediately after the entry of sporozoites into the human body when bitten by female anopheles? Explain. See, for this question, what you have to remember? You have to remember that this malaria, who causes malaria? Malaria is caused by Plasmodium. Falciparum. Not only the Plasmodium falciparum, the Plasmodium ovale is there, Plasmodium malariae is there, Plasmodium vivax is there. So, this causes malaria. For that, we require a vector. For that, we require a vector. And what is a vector? Vector is a female anopheles. It is a female anopheles, right? When a normal person is bitten by a Plasmodium, when a normal person is bitten by this female anopheles, this causes a transfer of this Plasmodium falciparum into their body. So, first of all, what happens is, the first organ which is affected, is liver. Right? Exact cycle is, first of all, these hepatocytes are affected, and later on it is transferred to blood. Later on it is transferred to blood. In blood, it causes destruction of red blood cells. Red blood cells. So, when infection occur in RBC, there is a release of hemazoin. Because of release of hemazoin only, we feel fever and chill. Right? Now, listen very carefully. Female anopheles bites a normal person carrying these Plasmodium falciparum. which can cause malaria when these sporozoids are released. So first of all, what happens is these sporozoids, these are released into the body, and first of all they get entered in blood. When from the blood they circulate for about half an hour and they are avoided by our immune system, they enter into the hepatocyte. What is the name of liver cell? Hepatocyte. From hepatocyte it migrates to blood, it causes destruction over there, number of RBC decreases, they releases hemozoin due to which we feel fever and chill, right. So it takes time before the release of hemozoin and the symptoms will appear. So symptoms appear after some time, right. Now come back to the question again. Why do the symptom of malaria not appear immediately after the entry of sporozoid into the human body when bitten by human anopheles, right? Is it clear, right? Let's talk about the second part. Give the scientific name of the malarial parasite that causes malignant malaria in case of human. So for this, the correct answer you have is Plasmodium falciparum. Plasmodium falciparum, it causes malaria. Which malaria? Malignant. Right? Is it fine? So note this down, I'll be back. So students, hope you all have noted down this. So this is the question number 39. So you should have a clear idea of the life cycle of this Plasmodium falciparum or Plasmodium vivax, anyone you can take. In this you have to explain the schizogony as per the liver cell or the hepatic cells are considered. In this you have to explain the schizogony of the RBCs as well. Pre-erythrocytic and the erythrocytic schizogony, both you should know. Where this sexual cycle occur, where this asexual cycle occur, this is also a very important question which can be asked. A direct question from the life cycle of this malaria can be asked in your examinations. So let's move on to the next part, that is a question number 40. So question number 40 indicates name and explain and giving reason the type of immunity provided to a newborn by the colostrum and the vaccination. Now let's have a look. So, to talk about immunity, what is the immunity? Immunity is capable or capability of a body to fight against a disease. Capability of body to fight against disease, right? Now first of all, let's discuss the antigen. Antigen are any foreign molecule which can evoke our immune system is considered as an antigen. Let's talk about antibody. Antibodies are the cells produced by B cell of immune system. These are produced by B cell of immune system, right? Is it fine? So antigen antibody concept, hope it is clear to you. There are different type of immunity. One is called as a active immunity, and second is considered as a passive immunity. Active immunity and passive immunity. So the active immunity means to say our body immune system is active and body is producing antibodies. Body is producing antibodies, right? This is called as an active immunity. Now, on the contrary, what do you mean by this passive immunity? Passive immunity, that means antibodies are given from outside. right?